Be talking the next aspect of glucose oxidation. So just say a quick hello everybody. Just tell me if audio video is clear. I'm Dr. Poonam Agrawal. Will be teaching you the TCA cycle today, which is the next aspect after the glycolysis and PDH complex has taken place. In the previous class, I talked to you regarding how the glucose is getting oxidized in the glycolysis in both aerobic and anaerobic state, producing you lot many ATPs. And then I told you the acetyl-CoA is generated in the PDH complex, right? So now today is the time when I discuss you the TCA cycle. And the acetyl-CoA, which is generated from the pyruvate, is going to enter in the TCA cycle, which is lying in the mitochondria matrix, right? So TCA cycle, if I am discussing, I need to tell you this cycle, all the enzymes of this cycle is produced in the mitochondria matrix, right? So it is the mitochondria matrix where this TCA cycle is taking place. And in multiple reactions, what is actually going to happen? That acetyl-CoA is completely getting oxidized. It's converted to carbon dioxide and water. And during this process, 10 ATP is generated from 1 acetyl-CoA. So you need to understand how the reactions of TCA cycle is taking place and what all steps the ATP is getting generated and in what form exactly, right? So when we talk about TCA cycle, you will see there are multiple reactions. And first of all, what happens? The acetyl-CoA is getting condensed with oxaloacetate. So when we are talking about TCA cycle, I write it here. The acetyl-CoA, which is generated from pyruvate oxidation, is going to enter in the TCA cycle, which is there in the mitochondria matrix. There are a number of reactions which are actually going to make a circle. So that is the reason it is called TCA cycle, otherwise called Krebs cycle, because the scientist Krebs discovered it first. This acetyl-CoA is 2-carbon compound, which is going to condense with 4-carbon oxaloacetate. Right? 4-carbon oxaloacetate is there in the TCA cycle, which is going to be the first substrate. And this is going to combine with this 2-carbon acetyl-CoA to make you citrate, which is 6-carbon compound. This citrate is converted to cis-aconitate, which is next converted to isocitrate. So this cis-aconitate is also 6-carbon and isocitrate is also 6-carbon. You need to know the flow of intermediates in the TCA cycle. So what is happening? First of all, citrate, then cis-aconitate, then isocitrate is getting produced. Now what is going to happen next is this isocitrate is going to get converted to oxalosuccinate, which is again 6-carbon compound. We write it in bracket like this because this intermediate is going to be there for very little time in the cycle. Now this is quickly getting decarboxylated and this will result in production of alpha-ketoglutarate, which is 5-carbon compound. This 5-carbon alpha-ketoglutarate is again undergoing decarboxylation, producing you succinyl-CoA, which is 4-carbon compound. And this succinyl-CoA is converted to succinate, which is converted to fumarate, malate, and this malate again is converted to oxaloacetate. So you need to learn this somehow, that 6-carbon compound, citrate, cis-aconitate, isocitrate, oxalosuccinate, all these are produced. Then you have only 1 carbon, 5 carbon compound in this TCA cycle, and that is going to be alpha ketoglutarate, right? And on the left side, if you see, these all are 4 carbon compounds which are generated. Even oxaloacetate is 4 carbon compound. So this is the flow of intermediates in the TCA cycle. And if we talk about various enzymes, the first enzyme is going to be citrate synthase. The second enzyme is going to be aconitase. The third enzyme is going to be isocitrate dehydrogenase. Isocitrate dehydrogenase. Isocitrate dehydrogenase. Here also we write isocitrate dehydrogenase. Then what is the enzyme complex which is going to act on alpha ketoglutarate? You can even guess. It is going to be alpha ketoglutarate dehydrogenase complex. This is a complex of enzyme. Just like your PDH complex of enzyme, which I told you in the previous session. So this particular enzyme complex is going to be alpha ketoglutarate dehydrogenase complex. Alpha ketoglutarate dehydrogenase complex. This enzyme is succinate thiokinase. Succinate thiokinase. And this enzyme is going to be succinate dehydrogenase. There are many enzymes. Initially, it will be probably difficult for you to understand and learn these enzymes, but slowly when you practice, you will learn all these enzymes properly. Succinate dehydrogenase is going to dehydrogenate the succinate and fumarate will be formed. And this next enzyme is going to be fumarase, which will add on one water at this step. And this enzyme is going to be malate dehydrogenase. Enzyme is going to be malate dehydrogenase. So citrate synthase, aconitase, you need to remember all of them. Citrate synthase, aconitase, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, succinate thiokinase, succinate dehydrogenase, fumarase, malate dehydrogenase. You have so many enzymes. And out of these various reactions in the TCA cycle, you must know only 2 reactions are single direction. And those 2 reactions which are single direction are citrate synthase and second one is alpha ketoglutarate dehydrogenase, which I have marked in the bold arrow like this. And other all reactions are going to be reversible fashion. All are reversible except 2 reactions which are irreversible. And these 2 irreversible may be MCQ to you. So we can write it here, the irreversible reaction. Irreversible reactions of TCA cycle. They are number 1, citrate synthase, and number 2, alpha ketoglutarate dehydrogenase complex. These are important MCQ. All other reactions are going to be reversible, and this is again very important for you to know which reaction is reversible and irreversible, because for the process of gluconeogenesis, which I'll be talking next, you need to have certain reactions of the TCA cycle as well. So it is important to pay attention to the direction of reaction, whether it is reversible or it is irreversible. This alpha ketoglutarate dehydrogenase complex enzyme is very similar to PDH complex of enzyme. You have seen in the previous session where PDH complex enzyme I discussed in detail, and I told you that PDH complex enzyme has got 3 enzyme and 5 coenzyme. Similar fashion, this alpha-ketoglutarate dehydrogenase complex also has got 3 enzyme and 5 coenzymes in it. When we talk about the gain of ATP, you must know the ATP is actually produced in the form of NADH and FADH2. There are 3 positions where the NADH is produced, and there is 1 position where 1 FADH2 is produced. So let's first talk about those steps where NADH is produced. So the number 1 step where the NADH is produced is the isocitrate dehydrogenase step, where NAD is going to be coenzyme, and that is going to accept the hydrogen from this isocitrate, and that NAD will be coming out in the form of NADH. So that's the very first point where you get 1 NADH. Then coming to this alpha-ketoglutarate dehydrogenase complex, at this position also 1 NADH is generated. It is just like PDH complex, where you saw that PDH complex when acts produces you 1 NADH. Last but not least, the malate dehydrogenase enzyme is going to produce NADH. So this is how we talk about the production of NADH at 3 positions. One is isocitrate dehydrogenase, another is alpha-ketoglutarate dehydrogenase, and third one is going to be malate dehydrogenase. So we write it somewhere here that the number of NADH produced is 3, and this 3 NADH can generate you 7.5 ATP, because 1 NADH produces you 2.5 ATP. So 3 NADH, which is produced in this fashion, is going to generate you 7.5 ATP. There is only 1 position where 1 FADH2 is generated, and that particular enzyme you must never forget. That's going to be succinate dehydrogenase enzyme. That's going to generate you 1 molecule of FADH2 here, because the coenzyme required for succinate dehydrogenase is FAD. So that is going to generate you 1 FADH2. And 1 FADH2 generates 1.5 ATP. So this is going to be 1.5 ATP. Now there is one place where the ATP equivalent GTP is getting produced. ATP equivalent GTP is getting produced, and you must know that particular step is the thiokinase step, where GTP is required, and that's converted to GTP. So this is 1 GTP which is produced in the TCA cycle, and this 1 GTP is equivalent of 1 ATP. So this fashion, if you see, the total number of ATP which is generated is 7.5 plus 1.5 plus 1 ATP. That is going to be total 10 ATP which is generated. So 1 acetyl-CoA is going to produce you 10 ATP, and that is so much of energy which is gained from 1 acetyl-CoA. You must know that here we are talking about the acetyl-CoA, which is coming from glucose pyruvate oxidation. You must additionally know that this acetyl-CoA may even come from amino acid metabolism. Pyruvate I already told you, which is coming from glucose. This acetyl-CoA may also come from fatty acid beta oxidation, which you will be learning later. So whatever the material is, whether it is amino acid or the glucose or the fatty acid, ultimately whenever we are going to get the acetyl-CoA, this is going to get utilized in this TCA cycle for the sake of production of 10 ATP. I just hope it is clear to you, all of you. So we have talked about the reaction, direction, enzymes, and various ATP production. And I have additionally told you the source of acetyl-CoA may be glucose, maybe amino acid, maybe fatty acid beta oxidation. So by this time, if you have any doubt, you can ask me. Hello, everybody. So please just put your queries in the chat box. If you have any doubt, I will solve your doubt then and there. Now, when we talk about TCA cycle, there are many enzymes which are having certain regulatory factors working for them. At times, those enzymes are getting activated, and some other time, those enzymes are getting inhibited. So we have certain enzymes in the TCA cycle which are under strict control of allosteric modifiers. And regarding that, I would like to show you one table which is talking about the various factors which are affecting the TCA cycle enzymes. So you can make a note that there are three enzymes. One is citrate synthase, another is isocitrate dehydrogenase, and third one is going to be alpha keto glutarate dehydrogenase enzyme. Alpha keto glutarate dehydrogenase enzyme. So these are the various enzymes which are under strict control. They are called regulatory enzymes. Regulatory enzymes of TCA cycle, which is a direct MCQ to you. Citrate synthase, when you talk about, this is getting activated by ADP and inhibited by acyl-CoA. Inhibited by acyl-CoA. Isocitrate dehydrogenase activated by ADP, inhibited by NADH, and also inhibited by ATP. When you talk about alpha ketoglutarate dehydrogenase complex, this is also having similar kind of control, where energy deficient state in the form of ADP is going to activate this enzyme, and NADH and ATP is going to downregulate this enzyme. So these are various regulatory enzymes which you must know are acting in the TCA cycle and controlling the rate of the reaction. Now, when we talk about the inhibitors of TCA cycle, we have three variety of inhibitors which are going to inhibit the TCA cycle reaction. So you must know them all, because very frequently they target you in your exam, that what all inhibitors are there and which exact enzyme is going to get inhibited by them. So in that context, you must know a good mnemonic, famous Ux. Famous Ux is a good mnemonic to remember the name of inhibitors in the TCA cycle and the corresponding enzymes which they are going to inhibit. So F stands for fluoroacetate, A stands for aconitase, and M stands for malonate. A stands for, this A is for arsenite, right? So let me make a little correction in this. This is arsenite and this is malonate. And these are the name of enzymes, 1, 2, 3. This is aconitase. This is alpha ketoglutarate dehydrogenase. And this is going to be succinate dehydrogenase. Succinate dehydrogenase, right? So why we are writing it like famous Ux? Because this helps you remembering that fluoroacetate is going to inhibit aconitase enzyme, right? You can make a small arrow like this here. And arsenite is going to inhibit alpha ketoglutarate dehydrogenase enzyme. You can make a small arrow like this. Malonate inhibiting the succinate dehydrogenase enzyme. You can make a small arrow like this. So this will allow you to remember the name of various inhibitors and their corresponding enzymes where they're going to inhibit the reaction of TCA cycle. And this is all very important for you, which you must know. Mishti, शांत हो जाओ. All right, so this is about the inhibitor of TCA cycle. So what we have seen, that how the reaction of TCA cycle is taking place and what all inhibitors are going to inhibit the TCA cycle at various steps. And I have told you the easy fashion in which you can remember the inhibitor and the corresponding enzymes which they are going to inhibit. Now you must know that TCA reactions are not dedicated only for the TCA cycle. There are many intermediates which are like going away from the TCA cycle, and there are many intermediates which are going to come inside the TCA cycle. Like for example, if I just take a red arrow and try to explain you that what is happening with citrate, that at times the citrate goes out for fatty acid synthesis, leaving the TCA cycle. Similarly, when you talk about alpha ketoglutarate, it may go out in the form of glutamate when amino group ammonia is added in this. Similar fashion when you're talking about malate, that also may go out for the process of glucose synthesis in gluconeogenesis. So these reactions which are extracting the TCA intermediate into various other reactions, and why to forget the succinyl-CoA, this may move to heme synthesis. So number of intermediates are going to leave the TCA cycle, going to go away in various other reactions, and these reactions which are emptying the TCA cycle of their corresponding intermediate are known as cataplerotic reactions. So I write here cataplerotic TCA cycle. We talk about two categories of reaction. One is cataplerotic reaction, which I was explaining you just now. And another is anaplerotic reaction, which I'll be explaining you now. So cataplerotic reaction are otherwise called emptying reactions, where the TCA cycle is being vacated of its intermediate that getting extracted in various other pathways. And anaplerotic reactions are just opposite. They are called filling up reactions, where various non-TCA reactions are providing the TCA intermediates, right? So non-TCA reaction provide TCA intermediate in anaplerotic reaction. So filling up reactions are anaplerotic reactions, where non-TCA cycle reactions are going to provide you TCA intermediate. And the reactions which are going to utilize the TCA intermediates into non-TCA pathway are called cataplerotic reaction. TCA intermediates used for non-TCA reactions. So this is how a lot of reactions are there which are going to utilize the TCA intermediate, and at the same time there are reactions which are going to provide the TCA intermediate. So it is a sort of balance between anaplerotic and cataplerotic reactions which determine the rate of TCA cycle. So this is all I wanted to talk to you regarding TCA cycle, where number of reactions are there, and we have to understand that TCA cycle intermediates are very much Mobile intermediates, they come into the TCA cycle, they go away from the TCA cycle. You have sets of anaplerotic, cataplerotic reactions. But the ultimate purpose of the TCA cycle is to provide ATP. That's entirely a different thing, that some of the reactions of the TCA cycle are getting utilized for gluconeogenesis as well, which I'll be discussing in the next section. So, but the main purpose of TCA cycle is catabolic reactions where the acetyl-CoA is undergoing catabolism. If any doubt, you can ask me at this stage. Otherwise, I will go to the next aspect where I will be taking a new pathway which is called gluconeogenesis, and will be explaining you in today's class how the process of gluconeogenesis produces glucose in the organ like liver, kidney, and very rarely GIT cell. So gluconeogenesis is a very important process where body organs are producing glucose. Of course, this is going to take place at the time when there is no glucose supply from the food. You must know this gluconeogenesis is an energy consuming process. So this process is not occurring every time. If you're eating food, this process will not be taking place. So this process takes place when there is fasting state, number one, and number two, insulin is not there. Lack of insulin. So always remember, whenever we are talking about glycolysis, we say that glucose must be available. The insulin must be available. But when we are talking about gluconeogenesis, which is just entirely logically a different pathway opposite to that of glycolysis, that gluconeogenesis is taking place only when the person is fasting, when the dietary glucose is not available. And number two, when the hormonal state is in such a fashion that insulin is not there and rather glucagon is present. Glucagon is present. So under these conditions, we have this pathway of gluconeogenesis, which is partly mitochondrial, partly cytosolic pathway. So I'm going to explain you how this gluconeogenesis takes place from pyruvate. In fact, when we are talking about the substrates which are going to produce glucose, there are only limited number of substrates which are capable of producing glucose by gluconeogenic reactions. First and foremost is the pyruvate. Then we talk about lactate. Then we see the glycerol, which is an alcohol which we consume in diet in the form of triacyl glycerol, is also capable of making you glucose. Then you have fumarate. Then you have glucogenic amino acids. All are capable of producing you glucose. So the basic fundamental approach is the understanding of glucose formation from the 3-carbon compound pyruvate. And you must know the major source of this pyruvate, which is gluconeogenic, is the alanine. Alanine, the amino acid. Amino acid. This is undergoing transamination to make you pyruvate. And whenever we are talking about the pyruvate producing the glucose, that pyruvate must come from amino acid only. How this pyruvate is making you glucose, that we need to understand. So for that, you must know that for gluconeogenesis, For, I write it here, for gluconeogenesis from pyruvate, all reversible reactions of glycolysis are used in reverse direction and irreversible reactions of glycolysis is getting bypassed. It's getting bypassed, right? So if you see, if you want to learn the gluconeogenesis, first of all, you must see the video where I have told you how the glycolysis is taking place. There you have beautifully seen that only 3 reactions are irreversible. All other reactions are reversible, and ultimately in the glycolysis, glucose is converted to pyruvate. So if I am talking to you about making of glucose from the pyruvate, I need to talk about that skeleton of glycolysis only in the reverse direction, where all the reversible equations are used in the reverse direction and irreversible reactions are getting shunted, getting bypassed. So that is what I am written here. For the gluconeogenesis from the pyruvate, all reversible equation of the glycolysis is used in the reverse direction and all irreversible reactions are bypassed by a new set of reactions. So if we follow that, you need to know the outline of the glycolysis first of all, where I told you glucose converted to glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-bisphosphate. This is breaking into 2, DHAP and glyceraldehyde 3-phosphate. I'm just writing the outline of the glycolysis, because unless until you know the enzymes of glycolysis and direction of various enzyme activity in the glycolysis, you will not be in a situation to exactly understand the process of gluconeogenesis. Glyceraldehyde 3-phosphate converted to 1,3-BPG, 3-PG, 2-PG, 2-phosphoglycerate, right? Then you have phosphoenol pyruvate. For detailed description of glycolysis, you can see the video which I have uploaded. In the previous session, I took your class for glycolysis. There I told you all these steps in detail. Then this phosphoenol pyruvate is converted to enol pyruvate, and which is next converted to keto pyruvate. This is how the glycolysis takes place, right? Now, if you talk about various enzymes, which you must know, this is glucokinase enzyme. This is phosphohexose isomerase enzyme. This is PFK1 enzyme. This is aldolase enzyme. Aldolase enzyme. This is going to be glyceraldehyde 3-phosphate dehydrogenase enzyme. Do you remember the enzyme here? This is phosphoglycerate kinase enzyme. This is mutase enzyme. This is enolase enzyme. And this is pyruvate kinase enzyme. The irreversible enzymes are pyruvate kinase, right? Number 1. Glucokinase, phosphofructokinase 1. This is how 3 enzymes are irreversible, and phosphohexose isomerase, aldolase, all this dehydrogenase, kinase, mutase, enolase, all are reversible. In fact, the enol ketone interconversion of pyruvate is also reversible. So if you're planning to produce glucose from this pyruvate, you have to follow the reverse direction where it will be converted to enolic form of pyruvate automatically, which next would like to get converted to phosphoenol pyruvate. So this kind of shunting will be required. Many reactions will be required to convert enolic form of pyruvate to phosphoenol pyruvate. So let's talk about that reaction in gluconeogenesis. How this enol pyruvate is converted to phosphoenol pyruvate. For that, I'm going to write enol pyruvate here. And we'll be explaining you how this phosphoenol pyruvate is produced. This enol form of pyruvate is there in the mitochondria, and this is going to get converted to oxaloacetate, which will be converted to malate. This malate will come out in the cytosol, will be converted to oxaloacetate, which next will be converted to phosphoenol pyruvate by decarboxylation. There is a carboxylation at this point where 3-carbon pyruvate is converted to 4-carbon oxaloacetate, and then there is a decarboxylation at this point in the cytosol, which is converting 4-carbon oxaloacetate to 3-carbon phosphoenol pyruvate. The enzyme which is converting oxaloacetate to malate is malate dehydrogenase enzyme, and even in the cytosol you have cytosolic malate dehydrogenase enzyme. This enzyme is very important, and this is called phosphoenol pyruvate carboxy kinase enzyme, which demands GTP for action. This enzyme is pyruvate carboxylase. Pyruvate carboxylase, which is demanding ATP for action. So this is how, whenever we are talking about pyruvate converting to phosphoenol pyruvate during reversal of glycolysis, we encounter so many enzymes, the pyruvate carboxylase. So that's a gluconeogenic enzyme that you can see by now. You understand this is gluconeogenic enzyme. You need phosphoenol pyruvate carboxykinase enzyme. That is also gluconeogenic enzyme. So by sets of these reactions, the pyruvate is converted to phosphoenol pyruvate. And next, what is happening? This phosphoenol pyruvate is converted to 2-phosphoglycerate by enolase action, because that's a reversible one. 3-phosphoglycerate mutase is also reversed, PG kinase is also reversed. All these reversible equations will take place, and fructose 1,6-bisphosphate will be produced. Now PFK1 reaction will be bypassed in the gluconeogenesis by fructose 1,6-bisphosphatase enzyme. Fructose 1,6-bisphosphatase enzyme. This is bypassed reaction, which is bypassing the PFK1. This itself will be a direct question to you. That what is the enzyme, gluconeogenic enzyme, which is bypassing the action of PFK1? You must know it is going to be fructose 1,6-bisphosphatase enzyme. The enzyme which is going to bypass glucokinase is going to be glucose 6-phosphatase enzyme, which is endoplasmic reticulum lumen enzyme. So This is how glucose is getting synthesized from 2-pyruvate. I again repeat it in brief that pyruvate need to take the reverse route of the glycolysis, but just because all the reactions of the glycolysis are not reversible, it is not easy for the pyruvate to just make a swift conversion to the glucose. Pyruvate need to bypass certain irreversible equations. The first one is the pyruvate kinase. The bypassing that pyruvate kinase is a multi-enzyme process, which I've explained you here. And then beyond that, enolase, mutase, all these glycolytic enzymes are going to be used up in the process of gluconeogenesis. Only the problem will be coming at the PFK1 step, which will be bypassed by fructose 1,6-bisphosphatase. Then phosphohexose isomerase will be used. Then glucokinase action will be bypassed by glucose 6-phosphatase activity. Ultimately, glucose will be formed. Right? So, this is how glucose is produced in gluconeogenesis. And now by this time you have known that the reversible glycolytic enzymes are common for glycolysis as well as for gluconeogenesis. But irreversible reactions of glycolysis catalyzed by glucokinase, PFK1 and pyruvate kinase are dedicated enzyme for glycolysis, which are being shunted by other reaction routes during the process of gluconeogenesis. I said that insulin must be absent if you want the glucose synthesis to take place. Rather, you must have glucagon. So regarding this, when I talk to you about this pyruvate carboxylase enzyme, which is very important enzyme for the process of gluconeogenesis, unless until it is there in the mitochondria matrix, you cannot even talk about the process of gluconeogenesis. In this regard, for this particular enzyme, I have a very important statement to say that gene of this pyruvate carboxylase enzyme is under suppression by insulin. So it means what? Whenever insulin is there, this enzyme is not produced because the insulin is going to have impact on the gene of this pyruvate carboxylase enzyme, which is getting suppressed, not allowing the pyruvate carboxylase enzyme to be formed. So unless until insulin is absent, you cannot think of the onset of gluconeogenesis. That is number 1 point. And number 2 point is, it is not mere the absence of insulin which is important for gluconeogenesis, for functioning of this particular enzyme. It is not the mere absence of insulin. You must know that number 1 is the lack of insulin which will allow the enzyme to be produced. And number 2, at the same time, you must have acetyl-CoE in the mitochondria matrix to stimulate, to activate, to allosterically activate pyruvate carboxylase enzyme. And if you want to know a little more, this acetyl-CoA is generated from fatty acid beta oxidation purpose. The fatty acid beta oxidation pathway is generating acetyl-CoA. And that is the reason every time they say that if you want to make the glucose in the liver must have background of fatty acid beta oxidation, because that fatty acid beta oxidation is going to provide acetyl-CoA, which will be activating the pyruvate carboxylase enzyme, which will be converting the pyruvate to oxaloacetate. Thereafter, the process of gluconeogenesis will begin. So these points regarding this pyruvate carboxylase activity, you must know. So this, I have told you how the pyruvate is making you glucose. If somebody is telling that during exercise muscle is getting lactate by anaerobic glycolysis, that time the lactate which is diffusing to the blood, how it is going and making the glucose in the liver. So you must remember this skeleton from pyruvate to glucose, and you can just add one line that, okay, if the starting material is lactate, first of all, that lactate need to convert itself to pyruvate by enzyme lactate dehydrogenase. And in this process, what is happening? The lactate is getting reduced to pyruvate. NADH will be required and NAD will be generated here. So this is how lactate will be converted to pyruvate, and from thereafter, pyruvate will be converted to glucose. So this is how lactate is also capable of making you glucose, but for that lactate need to, first of all, get converted to pyruvate. This is actually the part and parcel of the Cori cycle, which is very often a question to you, which otherwise is called is glucose lactate cycle. Yes, Ahmed, you're very much right. Acetyl-CoA is never gluconeogenic. That's very true. So that's true. Acetyl-CoA 2-carbon compound can never get converted to glucose. But what I have told you here, the role of acetyl-CoA in activating pyruvate carboxylase, that of course is very, very important. This acetyl-CoA itself is not converted to glucose, but it is important to stimulate pyruvate carboxylase enzyme, which can convert pyruvate to glucose. You understand? So there is a difference in these two statements, and your statements is absolutely fine, and that's a very much repeated MCQ, that anything can form glucose, but acetyl-CoA can never get converted to glucose. But still, we need acetyl-CoA for the purpose of activating pyruvate carboxylase enzyme, which then is converting pyruvate to oxaloacetate and initiate the process of gluconeogenesis. I hope it is very clear to you now. Yes, very good. So, saying this, now this is the time when I talk to you about Cori cycle, which otherwise is called glucose lactate cycle. This is a very important question, not only in prof exam, but also in the NEET, next, all types of exam. You must know what is happening actually in the Cori cycle. So regarding this, let me draw two organs here. This is liver. Of course, you recognize probably this is liver. And this is skeletal muscle. This skeletal muscle is exercising muscle, means the one which is like in rapid contraction. During this time, what is happening? The glucose is undergoing glycolysis, and due to contraction of muscle, the blood vessels are also getting contracted, and the oxygen supply is reduced in the muscle, resulting in anaerobic glycolysis, which means that we have generation of lactate from glucose. Generation of lactate from glucose. Right? Now what is happening? This lactate is going to come to the blood. This is blood. It's going to diffuse. No? When you take rest, then the pain is resolved. That's because of diffusion of lactate to the blood. Now the question is, what is going to happen to this lactate which is diffused to the blood? And the answer is, this lactate is going to go to the liver, and there this lactate is converted to glucose by process of gluconeogenesis, and this glucose being dumped in the blood again, which is getting utilized in the skeletal muscle. So if you see, these two organs are actually interconnected in this fashion. The muscle is generating lactate, and that lactate is like taken care of by liver. Taken care of by liver, and that's like converted to glucose. So glucose coming to blood, taken up by muscle, again converted to lactate, lactate coming to blood, taken up by liver, converted to glucose. It's a kind of cycle which is going on. This is called glucose lactate cycle, or otherwise called Cori cycle. This name is given by scientist Cori, who discovered this. So in this Cori cycle, the two pathway which is involved is, number one, the anaerobic glycolysis, and number 2, the gluconeogenesis. Yes, the two arms of this Cori cycle is anaerobic glycolysis, and number two, you have the gluconeogenesis, right? Gluconeogenesis, right? Very good. Yes, glucose 6 phosphatase is absent in muscle. That is the reason. Ahmed, you're absolutely right. Glucose 6 phosphatase, this particular enzyme is not there in the muscle. That is the reason skeletal muscle is not able to generate you glucose by gluconeogenesis. This glucose 6 phosphatase enzyme is there in the liver. So that is the reason liver is capable of making you glucose. Muscle cannot make you glucose. Because this terminal enzyme of gluconeogenesis is inherently absent in the skeletal muscle. Yes, Ahmed, you're absolutely right. Okay, alright. Any other question you can ask me? This is Cori cycle which you must know, right? और बताओ क्लास थोड़ी देर और चलेगी एशियन बॉय एट द रेट मेडिकोस ठीक है तो ये हमारी बात हुई gluconeogenesis की you must know all the substrates which is making you glucose. I've told you the example how the pyruvate is making you glucose. Then I told you that if lactate is there as a starting substrate, then how it is converted to pyruvate and then that is getting converted to glucose, right? If somebody asks you how this glycerol is making you glucose, glycerol we consume in our diet and that goes to liver. Glycerol is alcohol that can get converted to glucose. So this glycerol is converted to glycerol 3-phosphate. Glycerol is converted to glycerol 3-phosphate, which is next converted to dihydroxy acetone phosphate. Dihydroxy acetone phosphate. And this DHAP enters the glycolytic route where a reversal of this DHAP can produce you glucose. This enzyme is glycerol kinase and this enzyme is glycerol 3-phosphate. Dehydrogenase. This enzyme is glycerol 3-phosphate dehydrogenase enzyme. So glycerol also can produce you glucose, though in a very small quantity the glycerol is available for making of glucose. But if anybody asks you which step of glycolysis this glycerol is sending its intermediate, then you have to say DHAP. When you have 2 DHAP, 1 DHAP will be converted to glyceraldehyde 3-phosphate and both of them will combine to follow the reverse route in the glycolysis to produce you glucose. Now after learning this gluconeogenesis, I would like to talk about one more pathway which is a very small pathway but at the same time clinically very important pathway in human, and that is called fructose metabolism. We consume fructose either directly in the fructose form or in the sucrose form, and this fructose is getting predominantly utilized in the liver and some amount. I will draw a bold arrow here in the liver because major quantity of this dietary fructose is utilized in the liver and a small quantity of this fructose is utilized in spleen, placenta, testis, sperm. What is interesting here, the enzyme which is going to act on fructose in the liver cell is going to be fructokinase. And on the other hand, the enzyme which is going to act on fructose in other cell is going to be hexokinase. This fructokinase is converting fructose to fructose 1-phosphate, which is going to split into two, DHAP and glyceraldehyde. DHAP enters the glycolytic route and glyceraldehyde, after getting converted to glyceraldehyde 3-phosphate, is going to enter the glycolytic route. This enzyme is aldolase B. A and C will not act here. Aldolase B only will be acting here to break fructose 1-phosphate into two compounds, DHAP and glyceraldehyde. What ultimately you see that fructose ultimately is utilized in the process of glycolysis in the liver. Even in extrahepatic tissue, the same thing is happening, but the starting enzyme is going to be different, and that is going to be hexokinase, which is converting fructose to fructose 6-phosphate. And this is going to enter in the glycolytic route, because this is, of course, is the intermediate of glycolysis. Why we discuss this pathway in good detail is because deficiency of this fructokinase results in benign fructosuria, a condition where fructose is not metabolized. Of course, when very first enzyme of fructose catabolism is deficient, fructokinase enzyme is deficient, then liver is not going to use the fructose, and you will have a lot of fructose floating in the blood, which will come out in the urine, giving the name benign fructosuria. Benign fructosuria. It's a benign condition. Another deficiency is seen for the aldolase B, the deficiency of which will result in hereditary fructose intolerance. Hereditary fructose intolerance. Aldolase B deficiency, hereditary fructose intolerance, where this fructose 1-phosphate is accumulating in the liver, enlarging the cell. Liver will be enlarged. Baby will have hepatomegaly. Baby will have hypoglycemia as well. Baby will have high uric acid also in the blood. There are a lot of problems with this baby, and this is a very much lethal condition where the baby is not surviving beyond a decade or 5. So this is a very, very frequent question. They ask you hereditary fructose intolerance. What is the enzyme deficiency? You have to name it as aldolase B deficiency. So this pathway also you must know, the benign fructosuria and hereditary fructose intolerance. Please understand that this fructose intolerance is different from sucrose intolerance. So now the question is, what is sucrose intolerance? As the name implies, here the sucrose is not tolerated by the body. See, what is happening whenever we are consuming sucrose, we have lot of enzymes here, which is called sucrase enzyme, which acts on the sucrose and digests it into glucose and fructose. And these monosaccharides are going to get absorbed in the blood circulation. What is happening if the sucrase enzyme is deficient? That time, this digestion of sucrose into glucose and fructose will not be taking place, and you will have lot of sucrose in the GIT lumen which are not digested and not absorbed. That time, GIT bacteria will come and they will attack the sucrose. They will degrade sucrose into various gases like carbon dioxide, methane, etc. So, if you consume sweet food or the fruit juice or the honey, what is happening? The sucrose is not getting digested, and that's going to be there in the gut, where the GIT bacteria is going to convert that sucrose into various gases. And these gases are going to give flatulence, bloating sensation in the abdomen, diarrhea. This condition is called sucrose intolerance. The person is going to have GIT discomfort after consuming sucrose containing diet. This is different from fructose intolerance, which I was discussing you just some time back. This fructose intolerance is a cellular problem where during metabolism one of the enzyme is deficient, resulting in accumulation of fructose 1-phosphate within the hepatocyte, causing hepatomegaly. Okay? So this is how you need to learn the fructose metabolism. And I think this is enough for today. You have learned the TCA cycle, various reactions, inhibitors, etc. You have learned the process of gluconeogenesis, where I have told you how the shunting takes place. Then I have told you about the fructose metabolism, where you have seen how this fructose metabolism error results in two types of disease. One is essential fructosuria, another is fructose intolerance. Then additionally, I told you what is sucrose intolerance, where you have seen sucrase enzyme is deficient, and that is resulting in cramps, flatulence, and diarrhea. So thank you very much. If you have any doubt, you can ask me. Otherwise, I will stop the class now. Right? Thank you.